Broadcast is now starting. All attendees are in listen-only mode.
Hi everyone, welcome. I'm going to give us maybe just 30 seconds for people to continue logging in, and then we will kick things off. Thank you for being here. All right. Hi everyone. In the interest of time, I will go ahead and get us started. Welcome to the first session of the Acidification and Estuaries webinar series hosted by the Interagency Working Group on Ocean Acidification. My name is Courtney Witkowski. I work for the NOAA Ocean Acidification Program and also serve as the Executive Secretary for the Interagency Working Group on Ocean Acidification. The working group was established by legislation to coordinate ocean acidification research, monitoring, and outreach activities between federal agencies. We have 12 federal agencies who are active members. Last year, the working group identified increasing interagency coordination on acidification and estuaries as a priority for the federal government, which led us to hosting this webinar series. This series was specifically organized by NOAA, EPA, USGS, and NIST, but I want to highlight that other federal agencies certainly play an important role in estuary research, including NSF, BOEM, and NASA, among others. Our goal for this webinar series is to discuss the state of knowledge regarding acidification in estuaries, providing examples of information and applied science needs from managers and communities, and to discuss the unique perspectives and roles of federal agencies in the field. We hope to follow this up with a workshop in 2025 with the goal of identifying the highest priority scientific gaps that will allow agencies to meet public needs. And while we have an internal goal of coordinating between agencies to define swim lanes and opportunities for collaboration, we felt it was important to do so with the larger community interested in estuaries, including researchers, state governments, nonprofits, industry, educators, and others. You are all really important partners in executing this work and should inform our research priorities to ensure we are delivering the information you all need. We hope that you will participate in our future webinars. Our next one will be a deeper dive into the state of science around acidification and estuaries and will take place on Wednesday, October 23rd, from 2 to 3:30 p.m. Eastern Time. I'll share the registration link in the chat later on, and also we'll share our flyer for that at the end. Today, you'll be hearing from 3 speakers who will be setting the stage for future discussions. First, Dr. Janet Reamer will describe the differences between ocean and coastal acidification. Next, Dr. Chris Gobler will expand on research on acidification and estuaries. And lastly, Dr. Dwight Gledhill will speak on the various roles federal agencies have in estuarine acidification research and monitoring. We'll have time for questions and discussion at the end of our webinar, and I encourage you all to please enter questions in the chat or through the questions functions that GoTo has throughout the presentations. This is meant to be an interactive webinar series, so we also simply welcome you to share responses and feedbacks through comments, even if you don't have direct questions. And I'll now move on to introducing our first speaker. Dr. Janet Reamer is the Science Program Manager for the Mid-Atlantic Regional Council on the Ocean, known as MARCO. Her early work focused on nutrient chemistry, coincidentally in Chris Gobler's lab at Southampton College. Janet is a carbonate chemist by training and now a co-coordinator for MACAN, the Mid-Atlantic Coastal Acidification Network, and SOCAN, the Southeast Ocean Coastal Acidification Network, with experience in upwelling zones, coastal subtropical marshes, estuaries, and now coral reefs through her work with SOCAN. She is also now leading Marco's work on blue carbon and marine carbon dioxide removal, or MCDR, from the perspective of integrating acidification, MCDR, and blue carbon into Marco's water quality goals and projects. Janet, over to you.
Thanks, Courtney. And I just want to say it's a great honor and pleasure to be helping kick off this webinar series because our coastal estuaries are in desperate need of our attention right now. So just to kind of go over what we consider to be the differences between coastal and estuarine acidification and ocean acidification. When we're thinking about the open ocean, we think primarily about the absorption of CO2 into the water from the atmosphere due to fossil fuel burnings and the accumulation of carbon dioxide over the last few centuries since the Industrial Revolution. Additionally, when we think about sources of carbon dioxide and acidification in open ocean waters, we also want to think very hard about advection, not only from the coastal zone, but obviously from farther regions, but the upwelling is also of great importance in the open ocean in some regions. When we drift back into the coastal areas though, next slide please. We think not only about the absorption of atmospheric CO2, which we continue to have in the coastal zone, but additions of CO2, the primary drivers of pH, from terrestrial sources as well. So when we think about different sources of CO2 and decreased pH, first thing we're going to think about is estuarine inputs from riverine sources, because inherently freshwater brings lower pH. Ocean currents and tides can also transport acidified waters in some regions, depending on what the pH is of offshore waters. Sometimes it's actually more basic, and so the river or the coastal ocean end members actually help reduce acidification in estuaries in some instances. Additionally, stormwater runoff, septic leaching, and other point sources, especially effluent water from water treatment plants, can also contribute not only to eutrophication, which provides all of those excess nutrients for plant and algae and phytoplankton blooms, but the not having that water run through the aquifer and rather run off of impervious surfaces prevents the natural changes in pH that would occur. So again, depending on if the estuary is more heavily populated by obviously humans and all of our roads and highways and parking lots, then the amount of stormwater runoff is actually going to increase. Additionally, when we think about estuarine sources, we also think about controlling those nutrients, and improving water quality in the estuaries is probably one of the greatest ways that we can mitigate acidification. So as this slide highlights, if we can control the point source output, regulate fertilizers that would be used in terrestrial fields, and that leads to agricultural runoff of excess nutrients, then we can do a really good job of minimizing the impacts of acidification on estuaries. So it starts upland, not in the estuary itself. Next slide, please. Back one. There you go. So something that we think about when we're talking about coastal acidification versus the ocean acidification is framing and phraseology. So there's kind of two ways to think about acidification. So acidification is a verb. To acidify is the change over time towards the lower pH or aragonite saturation state due to atmospheric inputs of CO2. And this can actually be local or global scale. And the first thing that really comes to mind are these kind of iconic graphs at this point. The increase in atmospheric CO2 with the decrease in ocean pH. And that's something that we've been tracking, obviously, since basically since the Industrial Revolution. So on top of coastal acidification, all of our estuaries are also influenced by the absorption from the atmosphere. So to kind of compound that, we have this idea of an acidic event, which more you can think of it as a noun because it's something that's one thing that happens. So generally speaking, it's a short-term episodic acidic condition, which occurs on more of a local or a regional scale. And it can be caused by some episodic event, such as a hurricane. This example, and if you look at the middle panel and the top panel, the pH and salinity in the Chesapeake Bay and the Neuse River Pamlico Sound after hurricanes that passed, the hurricane actually kind of generates this episodic event where you have an influx of fresh water from all of the precipitation, but you also have an overturn of bottom water. And so especially in estuaries that have maybe longer residence time, like the Neuse River Pamlico Sound, you can have a greater accumulation of CO2 in the bottom waters, and then when the hurricane passes, turns over those bottom waters and brings all of that acidified water up to the surface. And then that can also generate a phytoplankton bloom, which you see in the Chesapeake example. And that bloom, after the CO2 is sucked out of the water from phytoplankton, it actually goes back into the water as those phytoplankton decay. So we have complex processes that are occurring in the coastal zone and in our estuaries, and each estuary is going to have different primary drivers of acidification because of the heterogeneity not only of space and time, especially along the east coast of the United States, but also just due to the different populations around those estuaries. So in short, we can't really consider estuaries on a whole. It's much more important to focus on individual estuarine drivers in place and time. Next slide, please. So when we think about estuarine acidification, we think about all of the important habitats that occur in estuaries. So this is just kind of a summary slide of different wetlands, oyster reefs, and of course, fisheries that depend on the protection of the estuaries. So coastal acidification is truly a water quality issue at heart. And controlling nutrient additions in the coastal zone will certainly help not only improve overall water quality, but it will help improve habitat quality in estuaries. Next slide, please. So what are we doing about acidification in our coastal zones? Well, Courtney mentioned the coastal acidification networks, and I think Dwight might be going into a little bit more detail about how the CANs were formed. So I'll kind of skip that part, but say that the CANs are congressionally mandated, which we're very happy about because it means that we have more of an established role in how we can help with combating coastal acidification. In a nutshell, our missions, and there are 7 CANs across the United States, and we're happy to welcome Carry CAN, the Caribbean Coastal Acidification Network, within the last few months. So our general goals and missions are to synthesize regional efforts, including data and different projects that are ongoing. We provide region-specific education and outreach to different organizations, whether they're already academic, federal, state. Specifically right now, I'll give an example of how MACAN is participating with state action planning. And then each of the different CANs convene work groups to address region-specific issues. We're very fortunate to have OAP backing us up and providing the space for us to also meet on a quarterly basis. And so the CANs really have a direct pathway not only to states, federal, and other nonprofits, but we also have a direct line to academia where we can participate and help synthesize all of this information. Next slide, please. So from a Mid-Atlantic perspective, a couple of weeks ago, we held a state-to-state collaboration meeting where all 5 of the Mid-Atlantic states participated. And so this table is kind of a snapshot of the type of action planning going on within the 5 states of the Mid-Atlantic and what each state is taking on within that action plan to help address coastal acidification. So I'm just going to go use New York as an example. In 2023, New York adopted a specific coastal acidification action plan, and it was led by the New York Department of Environmental Conservation. And it was the effort started in 2016, and the plan was adopted in 2023. All of the states also have begun to participate with the Ocean Acidification Alliance, which is a United Nations Foundation NGO. And OA Alliance actually helped us coordinate this workshop a couple of weeks ago. And I think it's really important to point out that, again, all of the states are addressing coastal acidification from a land-based pollution perspective. And I just want to highlight the great work that New York is doing also in exploring relevant marine carbon dioxide removal strategies, including nature-based solutions. So if you're interested in some more information on what Mid-Atlantic states are doing, I did provide an email address at the bottom, and I will also put that website address on the bottom. And I'll put that website address in the chat during the next speakers so that if you want to get more information on state level action planning, you can get that. So, thank you very much.
Thank you so much, Janet. Just 1 minute as we transfer over to our next speaker. Okay, I'm going to introduce Dr. Christopher Gobler while he pulls up his slides. Chris is the Endowed Chair of Coastal Ecology and Conservation in the School of Marine and Atmospheric Sciences at Stony Brook University. He researches global coastal change within 3 major themes: harmful algal blooms, interactions between climate change and eutrophication, and phase shifts in coastal ecosystems. He has a wealth of experience researching coastal acidification, and we're grateful to have him here today to expand further on this issue in estuaries. Chris, over to you. Oh, I'm not hearing you now.
Can you hear me now?
Yes, sounds great. Take it away.
Okay, great. Sorry for that little delay. Okay, well, thank you for the opportunity to speak, and great job by Janet giving us the introduction to the topic. I'm going to make essentially 6, or have information on 6 general conclusions, and so going through them, it'll be that coastal acidification is common in estuaries. It can vary across estuarine habitats. It can vary diurnally, tidally, and also seasonally. It can affect marine life. It combines with co-stressors to affect marine life. And we may not have the policies in place to protect marine life against these stressors. So just for perspective, of course, we're all aware of the global occurrence of hypoxia in coastal zones, and we're aware how that's driven by the overloading of nutrients and overloading of phytoplankton, and that primary production being broken down or respired microbially. In the process, of course, that takes up oxygen, but it produces CO2. And of course, the ocean doesn't care where the CO2 comes from. And so as that CO2 is added, just like it comes in from the atmosphere, if it comes in microbially, it also will acidify seawater. And so I'm going to just review some data from a few studies that were done with my doctoral student, Ryan Wallace, that I think gives some perspective on coastal ocean acidification. So in the first study we present, it's going to be focused, I guess it'll all be focused in the Northeast. And so just going to Long Island Sound, these are vertical section plots of dissolved oxygen May through November, with the far left being the most western area of New York City, and the eastern extent of Long Island Sound being out towards Rhode Island. And so what this shows is the annual development of hypoxia in the western bottom waters of Long Island Sound. And what you see is that that development co-occurs with the dropping of pH in the same exact regions. And when we did our own cruise, instead of relying on data collected through the EPA program for Long Island Sound Study, and we were able to also look at PCO2 and carbonate chemistry, and specifically targeting the end of summer. This is what it looked like. Now this only goes halfway through the sound, not all the way to the eastern extent, but still you get the sense. You have the development of the hypoxic region in the far west, particularly in bottom waters, that is accompanied by acidified waters that of course are high in CO2 and undersaturated with regard to aragonite. And the details are shown here, and so just for perspective, half of the data points in this cruise, the PCO2 is over 1,000 microatmospheres, and most of the water column at this time was undersaturated with regard to aragonite. And the pattern was similar in other estuaries. For example, when we looked at Narragansett Bay, in this case, the far left is the northernmost extent of Narragansett Bay near Providence, and the far right is the southernmost extent as it discharges more towards actually the Atlantic Ocean / Long Island Sound. And you see a similar pattern, even though we're looking at 3 different months in this case, June, July, August. The patterns are quite similar, and that is in the western extent, the more urbanized region, the development of bottom water hypoxia. That water is acidified, that water is high in CO2, and that water is undersaturated with regard to aragonite. And again, we're looking at thousands of microatmospheres of PCO2. Right, from this particular study was the direct effect of sewage discharge. As I gave the introduction, nutrient overloading can cause the overloading of organic matter, and that can be broken down to cause acidification. But for Ryan's dissertation, he also focused on the southwestern extreme of Long Island, looking at 2 different sewage treatment discharge points. And what he showed as he ran his continuous flow meters through there and was looking at using a PCO2 sensor, the levels of PCO2, what you see are these hot spots of CO2 with the levels being extremely high, again, in this case, over 2,000 microatmospheres in the discharge plume, and a very tight inverse correlation between the levels of PCO2 and the levels of oxygen in the water. And across all the locations they looked at in the study, you can see a very close correlation between the levels of oxygen and pH. Another sub-study within the work we did together was looking at different habitats, and specifically honing in on seagrasses, seagrass meadows, salt marshes, and areas dominated by macroalgae. And the first thing to assess was the levels of ecosystem metabolism. That is, on an annual basis, are these systems producing or consuming oxygen? And the salt marsh ecosystem was one that was you can see oxygen consumption. The algae system was more a producer of oxygen, as was the seagrass habitat. And we also looked at open waters, which were about even. And so with this as context for the oxygen dynamics, he also looked at, in this particular case, the dynamics of specifically on a diurnal basis, pH and oxygen. So these are time series done over an entire summer, actually. So each point is obviously one data point, and you can see it's multiple days over a 24-hour cycle. And what they show is, the easiest one, the open water habitat, things pretty steady. Well oxygenated, and pH hovering in the maybe 7.9 zone on average. Salt marsh, you can see the evidence of a diurnal cycle with very low levels of pH and oxygen at night, higher during the day. Even more exacerbated cycles in the macroalgal bed dominated region, whereas with seagrasses, you see the pH is staying fairly high and getting very high with high DO during the day. And then just looking at the carbonate chemistry here, consistent with the pH values, you see the higher levels of CO2 associated with the salt marsh ecosystem, also some high levels in the macroalgal bed, and low levels both in the open waters and the seagrass. And correspondingly, we've got very high levels of aragonite saturation in the seagrass and open water areas, and undersaturated within the salt marsh. And then a final study with Ryan to highlight, there'll be more after that, but just the studies with Ryan to highlight is looking at how algal blooms are affecting the dynamics of acidification and hypoxia. One thing that we did was a full cruise all around Long Island and through New York City. And you've already seen the area in western Long Island Sound that's high in CO2 and low oxygen. Another area that came up as high CO2 and low oxygen is Jamaica Bay. I already showed a little bit of data from that in the southwestern extreme of Long Island. And that's an area that seasonally over time does become acidified with high levels of CO2. You can see through the season that area expanding to cover the entire ecosystem by late summer, early fall. And consistent with that, those are times that overlap with low oxygen. And these conditions develop diurnally as well. So within this ecosystem, we did diurnal cruises from the afternoon through the night and into sunrise, and did those seasonally. And so you can see firstly the evidence of, upon daybreak, the breaking up of some hypoxia, and upon daybreak, and this is June, so daybreak's quite early, the normalization of pH. But seasonally, you see again the conditions get worse seasonally with regards to oxygen, pH, PCO2, and aragonite saturation. But you do see the persistence of our diurnal signal, and that is, at least in surface waters, conditions are somewhat normalized during the day, but at night you're getting the production of CO2, the consumption of oxygen, and the dropping of pH. Final thing that was done in Jamaica Bay was looking vertically. And for those of you who've ever been in New York City and flown to JFK Airport, this is JFK Airport. There's an old pier there. We were able to put in a vertically profiling device. And with that, over time, we were able to profile seasonally the changes in pH and DO. And this just shows an animation of that. But moving on to some of the details, we did this over 2 years, and combined, I think it was almost days worth of data. And what you see, firstly, is the development of ephemeral phytoplankton blooms in both years. The deployments you can see were from summer and fall. And those phytoplankton blooms injected into surface waters oxygen, and also were able to basify the water, or bring the pH of that water closer to normal. But what you notice is underlying these algal blooms are quite hypoxic and acidified waters, and you also notice that as the blooms decay and go away, the system sort of reverts to this hypoxic acidified condition. And so now just transitioning, of course, we know it's not just acidification in coastal zones, but we also concurrently have warm temperatures. I've already emphasized the hypoxia, and I'll mention briefly another stressor that co-occurs with all this are harmful algal blooms. And I've been known in the past to call these the four horsemen of the ocean climate change apocalypse. Maybe that's a little bit severe, but that these are all things that can threaten marine life. So we know what's happening with regards to temperatures, but sometimes in coastal zones, the temperatures extremes can be even greater. And so one of my former students, Stephen Tomasetti, focused on two of these stressors. I just want to highlight those. specifically temperature and low oxygen, because he was able to show using satellite-derived temperatures that just this century alone, that there's been warming of, getting towards 2 degrees over a 2-decade period in the area of eastern Long Island. This is just looking at summer periods, but that's what's most stressful for marine life. And these rates of increase are actually 3 times the global average. And when he looked to see the effects of marine life, they were significant. So he put out, for example, bay scallops, Orbopecten irradians, in multiple locations in the Northeast, both in Massachusetts and New York. He had one population that didn't survive at all, and he thought that was a failure in the experiment. But when he looked at the data, he saw that that collapse of the population occurred during a marine heat wave that co-occurred with hypoxic conditions. And in the lab study, he was able to, when he recreated these conditions, he was able to affirm that that was the cause of mortality. And then on the harmful algal bloom front, we know that's another stressor that's been expanding globally in recent decades, and the impacts in particular have been expanding. And sometimes that's linked to temperatures. This is a study where we showed that warming temperatures or changes in temperatures are expanding the season of some harmful algal blooms, in this case, Alexandrium. And in other cases, we've looked at acidification with regards to Alexandrium blooms, and have found that actually the areas, counterintuitively to what you think, phytoplankton are producing oxygen and consuming CO2. What we're actually finding, and this is just one example, is that an area with the highest densities of Alexandrium correspond and co-occur with the highest levels of PCO2. And this is something we've seen time and time again over multiple estuaries. Certainly are some exceptions, but we have seen that these areas do acidify through the season, and that's when you do get harmful algal blooms. So these stressors are co-occurring. And the question is, what are the implications of these co-occurring stressors for marine life? And I talked about that with a former doctoral student in this review paper here. In the interest of time, I'll just go on to some data on it. I think, in my lab over the years, we've really focused on larval stages of shellfish as a model organism to understand how they're affected by acidification and other stressors, because they can be quite sensitive. So this is a paper just looking at different levels of CO2 on 2 different species of bivalve larvae, and you can see a dose response. And you can see that these are levels we do see in estuaries, and they're decreasing the survival rates with each dose of CO2. And beyond survival, it also results in smaller larvae and juveniles as they develop. And then, finally, I think this is the last data slide. When we combine the acidification and a harmful algal bloom, we see these additive and synergistic effects. And so control groups here, fed and unfed, exposure to a harmful algal bloom here, exposure to acidification here, so depressed survival with acidification, but the worst outcomes when they get both stressors together. And a very final point is just a policy one. I know I'm out of time, so I'll just simply say that the Clean Water Act does regulate oxygen, but it doesn't consider other stressors. And so therefore, some of the guidelines for oxygen may not be as protective as we anticipate because there are these other stressors going on. So with that, I will end, and thank you for your attention.
Thank you so much, Chris.
You're welcome.
And we will now transfer over to our last presenter. And I'll introduce Dr. Dwight Phillips, the Deputy Director of NOAA's Ocean Acidification Program, where he provides critical leadership regarding NOAA's ocean acidification research portfolio. Dwight has a background in carbonate chemistry research, and he served as the chair of the Interagency Working Group on Ocean Acidification for this past year, and during that time, he helped to conceptualize this webinar series. Over to you, Dwight.
Excellent. Thank you very much. Assuming you can hear me?
Yes.
And you can see my slides.
Yes, you're good. Go ahead.
Excellent. Okay, well, thanks everyone for joining us. Real excited about this webinar series. And thank you both to Janet and to Chris for their opening presentations, which I think kicked us off exactly right. Let me see if I can get my presentation to work here. So I'm going to provide a pretty high level overview of the mission scopes of various agencies, not all the agencies that participate in the Interagency Working Group on acidification, but a few prominent ones where estuarine acidification is probably most relevant. And we're going to look at their missions. We want to look and see where they may overlap and intersect with other agencies and get a sense of where they're working and what they're thinking about going forward. And we're hoping that this might be a vehicle by which we can improve our coordination and leverage. So a big thank to Cheryl, Kim, and Regina for contributing slides to this presentation from their respective agencies. So I'll start with NOAA, my agency, most familiar with, and I'll just lay out for you kind of very broadly where we think estuarine acidification might intersect with our mission space. So NOAA's mission is revolves around science, service, and stewardship. Specifically, the agency looks to understand and to predict changes in climate, weather, ocean, and coast. And those coasts do include estuarine systems. We then are looking to share that knowledge and insight with others, particularly those engaged in decision. So we want to provide decision support, and decision support ideally that aims to help to conserve and manage coastal marine ecosystems and resources. So NOAA is relatively unique in that we have federal legislation that explicitly calls on this agency, NOAA, to engage in ocean acidification. It does call on a broad array of other agencies through the IWG to participate in the IWGOA, but it actually creates a program within NOAA, the Ocean Acidification Program, to help to not only invest in the research around acidification, but also to help to coordinate and keep track of a lot of the other science that's happening across our own agency, NOAA, on ocean acidification and other relevant areas. So that's the Federal Ocean Acidification Research and Monitoring Act, which I think maybe Janet spoke to. And it calls on NOAA to engage in research that explores not just the effects of acidification on ecosystems, but importantly, the socioeconomic consequences of those impacts. And it engages in all this research in alignment with a strategic plan, which was compiled and put together by the Interagency Working Group. So I did an attempt to list all the many external partners that we work with. I do think probably the most prominent external partners outside of the various agencies that we work alongside would be the regional IOS associations, which Janet, I think, laid out more eloquently than I possibly can. And many of those regional IOS associations currently include a coastal acidification network, which she spoke to, in an effort to try to provide some translation of the science to local stakeholders and to ensure that we're understanding what science questions are coming from those communities to inform the science that we're looking to invest in at the agency level. And of course, our cooperative institutes, which again, this allows for cooperative agreements for us to invest in academic partners that are engaged in science that's relevant to the agency mission. So in general, we have been engaged, at least our program has been engaged for more than 10 years now on looking at a whole transdisciplinary portfolio of science. So we certainly do monitor, a lot of monitoring, that's about half of our portfolio investment, but we also do a lot of examination of species sensitivity and increasingly, more and more, looking at the intersection at human dimensions. Okay, so just a very broad overview of some of the science that we've examined to date that's particularly relevant to estuarine science. There has been a fair amount of competitive funding opportunities that have come out of the agency, not just from our program, OAP, but there's been others from the National Estuarine Research Reserve System, particularly the National Sea Grant College Program has invested a lot of resources towards investigating species sensitivities in estuaries on acidification. And then IUS itself has made some investments on this front. And that research has encompassed a broad array of looking at not just the biology and the chemical and ecological responses in these systems, but looking at that multi-stressor aspect that Chris was speaking of. This is increasingly more so. So we're not just looking at acidification, but acidification plus temperature, plus hypoxia, plus nutrient loading. And more recently, there's a lot of work that's emerging on the front of mitigation through MCDR or even local adaptive strategies that can be employed to accommodate or mitigate against acidification. So I've shown here kind of a collage of various activities that we are engaged in. In the middle here, I'll show we have quadrennial coastal ocean acidification surveys that provide sort of the end member ocean condition states that can be used to inform estuarine model systems. And that's been an endeavor that's not just involved these coastal cruises, but also underway autonomous vehicles and systems. And then, of course, a whole array out of the NOAA, the National Ocean Acidification Observing Network, mooring time series stations. To date, there's not been a tremendous amount of those time series stations deployed in estuaries, but recently we made attempts to put one in the Chesapeake Bay at First Landing, and it's monitoring the dynamics that Chris spoke a lot about. And yeah, the values of CO2, even at First Landing, which is a relatively oceanic setting, are considerably more dynamic than what you see in the ocean environment system. We're using this data increasingly to inform routine products such as JScope, which provides seasonal outlooks that includes outlooks of bottom water pH in the Puget Sound on the West Coast. And more recently, we fund the routine product delivery out of VIMS that delivers 2-day forecast and nowcast of acidification conditions within the Chesapeake Bay. So some of the challenges I think that we're confronted with, which probably aren't unique to our agency, but monitoring within these systems is complicated by the fact that they're so incredibly heterogeneous, and we can't put an observing asset every square foot. So we have to rely a lot on local monitoring activities that are already in place. A lot of water quality monitoring is happening, some of it from NOAA and some of it from our partnering agencies, but acquiring good quality carbonate chemistry measurements within these systems does require some additional capacity investment and some technical assistance. So the actual number of analytical facilities at the local level that can run carbonate chemistry is relatively limited. And so we'd like to think about how we can improve that going forward. And many times that might just be training the people that are already engaged in water sampling on some of the new technologies that may be available, or even just training them on some of the old approaches that they may not be familiar with. The other challenge we have within estuarine systems, which we're hoping that this webinar series and this workshop will have to resolve, is just the delineation of swim lanes between the agencies within estuaries, because they are a bit of a tragedy of the commons. Everybody's in there, and we're not always sure what the left hand's doing. So that is clearly a role for the interagency working group, is to coordinate and understand and to leverage rather than duplicate. And data quality standards, I think, remains a challenge. So there are long-term time series in some of these systems of pH. Oftentimes they're based on NBS buffers that and other approaches which we may not fully understand the uncertainties around those. So important research questions, and this is just out of my own head, that may not actually fall within the window of NOAA's mission scope. So I'd like to ask the question about where we are in our dissociation constants for the carbonate system, for example. We have it very well carefully constrained in oceanic settings and freshwater systems, but in salinity dynamic environments, I think that can be complex to understand. Establishing a historical baseline by which to reference what today's state is all for the eye of the beholder. Clearly, you could look at what the estuary might have been in 1970, That was hardly much of a baseline. Most of these systems were occupied by considerable oyster reef habitats prior to colonialization. So our understanding of what an impaired estuary might be challenged by the historical context of what these systems probably once were. And then I think you'll see this in several of the agency slides, is trying to disentangle specifically what we mean by anthropogenic carbon in an estuary system is something that we want to look at carefully. So switching over to the Environmental Protection Agency, I believe estuarine acidification does intersect considerably with several of the mandates under the EPA, specifically under the Clean Water Act. The EPA implements regulatory and non-regulatory programs to protect water quality. One would assume that might include carbon chemistry. Combating nutrient pollution, which, as we've seen in the previous talks, is an important driver of coastal acidification, and to address climate change impacts like acidification. EPA administers permitting programs, which is critically important, particularly as we start venturing into this emerging science of NCDR. You're trying to permit against scientific field, which probably has fairly limited scientific understanding at this time. So trying to keep an open dialogue between the agency and the science they're supporting and the permitting that the EPA has to oversee is critical. So Cheryl points out here, estuaries acidification can be exacerbated due to local watershed-based activities and inputs of nutrients that cause hotspots and seasonal episodes. And I think both Janet and Chris communicated that excellently in their talks. They partner with states, tribes, national estuarine program sites, NOAA, of course, USDA, and universities. And I think I've already spoken to the permitting. So the kind of science to date that they've been engaged in is supporting the National Estuarine Program sites for monitoring acidification in estuaries, piloting OA indicators for national coastal acidification assessments, evaluating biological and ecological impacts. This looks very similar to some of the work that we've been engaged in at NOAA. Observing estuarine acidification responses to aggressive sewage treatment upgrades, developing simulation models to evaluate nutrient-enhanced acidification in estuaries, and monetizing the economic impacts of ocean and coastal acidification and temperature on shell fisheries in the US and Canada. Some of the challenges, well, resources are always a key challenge for all of the agencies, but in particular in the EPA, there's actually a fairly limited cadre of staff within the agency itself that are working on acidification aspects. And their monitoring programs tend to be operational for the most part. And so trying to find off-the-shelf sensors that are suitable and affordable for the water quality monitoring that the EPA is engaged in remains a challenge. So important research questions they've identified are distinguishing the role between global CO2 and local watershed drivers. I think we've seen that in the earlier talks, and I think we mentioned that for NOAA. Understanding the pace of biological acclimation and adaptation relative to the rates of change in estuarine systems. That's $1 million question. Clearly, NOAA's been engaged trying to look at acclimation and adaptation more recently, and the rates of estuarine acidification are oftentimes quite faster than what we see in the open ocean environment. And looking at the relations between nutrient pollution, acidification, and harmful algal blooms, which seems to be a common issue for many of the agencies. So the U.S. Geological Survey, mission and role for USGS includes natural hazards mission area, specifically the Coastal and Marine Hazards and Resource Program. They look to examine the impacts of acidification on seafloor, chemical erosion. As you can imagine, if you've got a karst environment, maybe that might be, the erosional rates of that might be accelerated by acidification, and that actually has knock-on effects to elevation change and related coastal hazards. Watershed mission area, looking at next generation water observing system, which I think Kim Yates has looked at particularly. Examining the impacts of fresh water delivery, changes in salinity, water temperature, and water quality constituents that contribute to coastal acidification. These riverine inputs, in many cases, into these systems is far outside the purview of NOAA in many cases, and it's really critical that USGS continues to keep these aspects as a high priority so that we have the boundary conditions necessary to model the estuarine systems and to get a better understanding of how best to forecast acidification dynamics. So ecosystem mission area environmental health program. And this speaks to their interest around HABs, which we saw for EPA and also for NOAA. They partner with, well, the EPA National Estuarine Programs, NOAA IUS again, National Park Service, which NOAA does as well. In particular, I know Kim Yates has been working closely with the Florida Keys National Marine Sanctuary. Oh, I lost my slide deck. Some of the NOAA laboratories like NOAA AOML. So USGS partners to address priorities, the USGS Ecosystem and Water Mission Area programs. Some of the activities that they're engaged in, there's some nice illustrations here. I think that's the time series station that Kim Yates has worked on for maybe more than 10 years now, located off the USF Port Station. That monitors carbonate chemistry dynamics, I believe both at the surface and in the subsurface, which is quite valuable. Doing discrete water sampling analysis, which allows you to do not only carbon chemistry analysis, but cell counts and looking at toxins. And their work on HABs, which again, that's pretty common across the agencies. Some of their challenges, again, capacity, funding, no surprise there. Prioritization is probably a real challenge within USGS, ensuring that acidification is recognized as an important issue within the agency. Important questions include, what's the impact of acidification on seafloor chemical erosion, like I mentioned previously? What is the impact of freshwater delivery, changes in salinity? So this is all of the upstream impacts of, both freshwater and its constituents to carbon dynamics in the estuary itself. And then understanding the effects of acidification and coastal acidification in the context of multi-stressors on HAB toxin production. That again rings like a fairly common theme. So all of the research and science that those agencies and the other agencies are engaged in acidification rests a lot on us being able to intercompare data. And much of that is stood on the shoulders of a single laboratory at Scripps, under the charge of Andrew Dixon for many years now, in delivering the community certified reference materials. So we as an IWG have been looking to NIST to help us on this front and to try to ensure that we have some certification process in place to certify other laboratories that could produce certified reference materials. And we think that falls squarely within this mission space, which is to promote measurement science, standards, and technology to enhance the productivity, facilitate trade, and improve the quality of life. Important research questions they're examining are developing NIST measurements for services to value assign total dissolved inorganic carbon and total alkalinity in seawater. And those measurements will include stated traceability and uncertainty budgets. And they're also continuing research on traceability of pH in seawater. So the challenge is that they currently don't have any direct funding for acidification work within NIST. So again, this is a priority challenge within that agency. And the bulk of the efforts today have been funded really by CO2 removal allocations. So this might be an opportunity for the emerging field of MCDR to maybe help double down and bolster the requirements we have for NIST to serve in this capacity. And yeah, I think I'll end there. So hopefully that was a whirlwind tour of the various agencies and what they're involved on. And as you can see, there's a lot of similar areas of science that I think we can better coordinate on. So thanks for your time.
Great, thank you so much, Dwight. And we have about 5 minutes left for some Q&A, and I'll invite Jana and Chris to turn their cameras back on as well. Want to run through a few of the questions we received from the audience first. Jana, I'll direct this first one to you. We received a question about if we can get Pennsylvania to join MACAN. They know that there is a tidal shoreline in the Commonwealth about 50 miles worth with Philly right in the middle of it.
Well, I believe that Kirsten Wakefield is also listening to the webinar today as well, who's my co-coordinator with MACAN. And we'll talk about it at our next meeting to see how we can potentially induce Pennsylvania. Just noting that Pennsylvania has a Sea Grant program, so should be hopefully an easy fit, because the Susquehanna River is one of the most prominent drivers of upper Chesapeake Bay acidification. All of the nutrient runoff, especially from Amish farmlands, which I can say I can see one out of my office window this morning. So
Great. Thanks, Janet. And I'll next turn to the question that asks, riverine dynamics seems to be crucial to understanding nearshore carbon cycling and acidification. What is the current avenue for supporting this kind of work? Chris, do you want to speak to that?
If it's support, I turn it over to Dwight.
Yeah, so I think this is where we run into the strategy of the commons that I hope this webinar series is going to try to overcome and I think if we have a better understanding of precise swim lanes and who's on first, I think that will help us better delineate how we can, if we can't increase the funding, at least we can do it in a way that makes it more impactful in a more coordinated fashion. So, there's any number of agencies that are currently supporting this kind of work, but whether it's at the level that it needs to be, I think is something, it wouldn't surprise me if we find that it's not sufficient. So I think part of this effort here is to try to help us develop the narrative we need to really recognize the importance of those kinds of observations and all of the knock-on benefits it would have. So
I'm going to jump in here and just say really quickly, I think, Dwight, you said it, leveraging and leveraging state-based research will also really help the overall picture of acidification along all of our coastline, because states do have robust monitoring capacity. It's just how are we putting that information to use right now? And we're not doing the best that we could be doing. So I think really engaging with states and using state level data and incorporating that into our federal monitoring plans.
Great. I'll throw another question out there. Someone asked, what are some of the benefits of measuring atmospheric CO2 in estuaries on a 24/7 basis in relationship to collecting estuarine water samples to study OA? Any reaction?
It's atmospheric. Janet, I think you'd be the right person on that front. But that's what they're asking.
Well, I think that right now, where our research stands is that, yes, we are still measuring atmospheric CO2 and trying to tease out how much atmospheric CO2 really impacts estuaries. My guess, and I would say educated guess, is that it really, the atmospheric component of dry estuaries is probably shadowed by the episodic events, anthropogenic, well, terrestrial anthropogenic processes are probably overshadowing. And Wei-Jun Tsai at the University of Delaware has done a lot of work in estuaries, and a lot of the early work that we are still building on today. I think we kind of took a period of time and skipped over estuaries, and we're really focusing on the open ocean, but we are coming back into estuaries now with more rigor and fervor than we have in the last 10 years.
can I just follow up on that too? And I think Janet made this point in her talk. All estuaries are local, right? So each estuary has to be treated in its own context. So estuary systems like Puget Sound are heavily influenced by upwelling, which contains a considerable amount of anthropogenic CO2 in those deeper water masses. So you have to go estuary by estuary to figure out what specifically, the relative role of atmospheric input is. And it also where you're looking in the estuary. So even in the Chesapeake, if you're looking out on the far edge of the Chesapeake, there is a considerable amount of anthropogenic DIC that's on that system. So as you go further up into the streams, for sure it changes. So that's part of the challenge.
Thanks, Dwight. We're at 1 o'clock, so I'm going to have to cut us off, but just want to direct everyone's attention to the chat for 2 links I shared. First is a registration link to join our next webinar on October 23rd. And then we unfortunately didn't have time to get to all of our audience questions, but I wanted to point everyone to a website called the oainformationexchange.org. If you're not already a member, you can create a free account very easily, but this is an online platform that is used for discussion of all kinds of things ocean acidification, and we will work on the follow-up post there with answers to the remaining questions. So I encourage you to go to that website, make an account so you can follow along for further discussion. And with that, I want to once again thank our speakers for their time and expertise, and thank you all for joining. Have a great day, everyone.